Welcome back, class. My name is Silvana O'Coney and today's lecture is lecture 6, the fungi. In this lecture we'll introduce the group of eukaryotes called fungi. We'll look briefly at their distribution, importance, structure, nutrition and metabolism, reproduction, and the 8 major fungal subdivisions. Let's start with an overview on fungi. Fungi are widely distributed and they're found wherever moisture, the appropriate temperature and suitable nutrients are present. They exist primarily as filamentous hyphae, and a massive hyphae is referred to as a mycelium. Just like some bacteria and some protists, fungi secrete enzymes called exoenzymes into the environment to digest insoluble matter. Then they absorb the solubilized nutrients. Most fungi have 2 reproductive structures: sporangia, which form asexual spores, and gametangia, which form sexual gametes. We use the term fungus, which comes from the Latin fungus, meaning mushroom, to describe eukaryotic organisms that are spore-bearing. They are chemo-organoheterotrophs with absorptive metabolism. They reproduce sexually and asexually, and they belong to the kingdom fungi within the domain eukarya. Sometimes the fungi are referred to as the true fungi or eumycota, which comes from the Greek meaning true fungus. Here's a slightly different version of that definition. Fungi are eukaryotic organisms that are spore-bearing, have absorptive nutrition, lack chlorophyll, and reproduce sexually and asexually. And you'll note the inclusion of lack chlorophyll. And this is just one of the major differences between kingdom fungi and kingdom plantae. Scientists who study fungi are called mycologists, and the study of fungi is called mycology. The study of fungal toxins and their effects is referred to as mycotoxicology, and diseases that are caused by fungi are referred to as mycoses. Fungi are primarily terrestrial organisms, although a few are found in freshwater or marine environments. They have a global distribution from polar to tropical regions, and while many are pathogenic in plants or animals, there are many that form beneficial associations with other organisms called mycorrhizae. For example, most plant roots form symbiotic associations with fungi, these mycorrhizal associations. Fungi are also found in the upper portions of many plants and can actually affect plant reproduction and also palatability to herbivores. Another example are the lichens. These are a symbiotic association of fungi with either photosynthetic protists, such as algae, or cyanobacteria. Now let's think about the importance of fungi, and you can probably imagine, with well over 90,000 fungal species described so far, that they're fairly important both in beneficial and harmful ways. Along with the bacteria and a few other organisms, fungi act as decomposers, a role of enormous significance. They degrade complex organic substances in the environment to simple compounds, and therefore carbon, nitrogen, phosphorus, and all the other critical elements are released from dead organisms and made available to other organisms. Fungi, especially the yeasts, are essential in industry, particularly in industrial fermentation processes, including the making of bread, wine, and beer. But they're also important in making some cheeses, soy sauce, in the commercial production of many organic acids, such as citric acid, in the production of some drugs, such as cortisone, and in the production of many antibiotics, including penicillin. Fungi are also important research tools, particularly in terms of studying fundamental biological processes. Fungi are used by geneticists, biochemists, biophysicists, and microbiologists. The yeast Saccharomyces cerevisiae is the best understood eukaryotic cell. It's been a model organism in the study of cell biology, genetics, and even cancer. On the other hand, fungi are a major cause of disease. Plants tend to be particularly vulnerable to fungal diseases because fungi can invade plant leaves through their stomata. Over 5,000 species can attack economically valuable crops, garden plants, and even wild plants. Fungi also cause many diseases of animals, including humans, and in fact about 20 new human fungal pathogens are documented every year. This table lists just some of the many diseases of animals caused by fungal toxins, otherwise known as mycotoxins. A mycotoxicosis is a poisoning caused by a mycotoxin. In this photo, which is a scanning electron micrograph at times 1000 magnification, we can see a young fungal mycelium forming over the stomata or stoma of a plant leaf, ready to penetrate. We'll now look at the structure of fungi. The body or vegetative structure of a fungus is called a thallus, plural thalli, and the thallus varies enormously in complexity and size, ranging from a single-celled microscopic yeast to multicellular molds, macroscopic puffballs, and also mushrooms. Some different fungal thalli are pictured here. In A, we have the multicellular common mold penicillium growing on an apple. In B, a group of puffballs, lycoperdon, growing on a log. And in C, of course, we have a mushroom. And a mushroom is made up of densely packed hyphae that form the mycelium or visible structure, that is the thallus. The fungal cell is usually encased in a cell wall of chitin, and chitin is a strong but flexible nitrogen-containing polysaccharide consisting of N-acetyl glucosamine residues. Most fungi can be broadly classified as either yeasts or molds. A yeast is a unicellular fungus that has a single nucleus and reproduces either asexually by budding and transverse division or sexually through the formation of spores. Each bud that separates from the mother cell can grow into a new yeast cell, and some group together to form colonies. Generally, yeast cells are larger than bacteria. They vary considerably in size, but most commonly are spherical to egg-shaped. They lack flagella, but they possess most of the other eukaryotic organelles, as pictured here. The molds are filamentous fungi. The thallus of a mold consists of long, branched, thread-like filaments of cells called hyphae, singular hypha, and these hyphae form a mycelium, which is a tangled mass of hyphae. In some fungi, some groups of fungi, the cytoplasm can stream directly through the hyphae uninterrupted by cross walls. These hyphae are called cenocytic or aseptate hyphae. The hyphae of other groups of fungi contain cross walls called septa. And the septa or septum, which is singular, contains either a single pore or multiple pores, and this enables the cytoplasm to move from one region of the hyphae into another. These hyphae with cross walls are termed septate. Hyphae are composed of an outer cell wall and an inner lumen, which contains the cytosol and organelles. A plasma membrane surrounds the cytoplasm and lies right next to the cell wall. So this filamentous nature of hyphae results in a large surface area relative to the volume of cytoplasm, and this makes adequate nutrient absorption possible. In this photo to the left, you can see the white hyphae that composes the fungal mycelium, and it's forming this macroscopic mass growing in and on the soil. To the right in A, you can see a drawing of cenocytic or aseptate hyphae. In B, we have a drawing of hyphae divided into separate cells by the presence of cross walls or septa. In C, an electron micrograph at 40,000 times magnification of a section of Dreschlera showing wall differentiation, that is the formation of a cross wall and a single pore. In D, we have a drawing of a cross wall, but this one has multiple pores. Many of the fungi, especially those that cause diseases in animals, are said to be dimorphic. That is, they have two forms. Dimorphic fungi can change from the yeast or Y form in the animal to the mold or mycelial form, otherwise known as M, in the external environment. And they do this in response to changes in various environmental factors. For example, nutrients, carbon dioxide, redox potentials, and temperature. This shift from Y to M or M to Y is called the YM shift. In plant-associated fungi, the opposite type of dimorphism exists. That is, the mycelial form occurs in the plant and the yeast form in the external environment. Let's turn our attention now to fungal nutrition and metabolism. Fungi tend to grow best in dark, moist habitats where there's little danger of desiccation, but they are found wherever organic material is available. Most fungi are termed saprophytes, that is, they secure their nutrients from dead organic material. Like many of the bacteria and protists, as we said earlier, fungi will release hydrolytic exoenzymes into the environment, and these enzymes digest large external substrates. The fungi can then absorb the soluble products, and this is a process that sometimes is referred to as osmotrophy. Fungi are chemo-organoheterotrophs, and they use organic compounds as a source of carbon, electrons, and energy. Glycogen is the primary storage polysaccharide in fungi, and most fungi use carbohydrates, preferably glucose or maltose, and nitrogenous compounds to synthesize their own amino acids and proteins. Fungi usually are aerobic. Some yeasts, however, are facultative anaerobic organisms, and they can obtain their energy by fermentation processes. Many of these fungal fermentations, of course, are of industrial importance, such as the production of alcohol in the manufacture of beer and wine. Obligate anaerobic fungi are found in the rumen of cattle. Now let's take a look at reproduction. Reproduction in fungi can be either asexual or sexual. Asexual reproduction is accomplished in several different ways. The first method of asexual reproduction is called transverse fission. During this process, the cell undergoes mitosis and divides into two cells by a central constriction and formation of a new cell. Mitosis may also result in budding, which is very common in the yeasts. During budding, a smaller daughter cell forms off the larger mother cell. But the most common form of asexual reproduction is spore production. And this also occurs through mitosis. There are several different types of asexual spores which are formed, and we'll look at those different forms on the next slide. Sexual reproduction in fungi involves the fusion of compatible nuclei. This is followed also by the formation of spores, but these are referred to as sexual spores rather than spores that are formed through asexual reproduction. Here are some of the main examples of sexual spores: zygospores, ascospores, or basidiospores. Here we have a diagrammatic representation of asexual reproduction in the fungi and some representative asexual spore types. In A, we can see an example of transverse fission occurring. In B, we can see a hypha that is fragmenting, that is by the separation of hyphae through splitting of the cell wall or splitting of the septum. And this fragmentation forms cells that behave as spores, and these cells are called arthrospores. In C, we can see the formation of chlamydospores. These are cells that are surrounded by a thick wall before separation. In D, we can see a sac called a sporangium, and within a sporangium are spores developing, and these are referred to as sporangiospores, and this occurs at a hyphal tip. In E, we can see that the spores are not enclosed in a sac but produced at the tips or the sides of the hypha, and these are termed conidiospores. And then in E, we have spores that have been produced from a vegetative mother cell by budding, and these are called blastospores. So as we said earlier, sexual reproduction involves the fusion of compatible nuclei. And this can occur as the result of fusion between haploid gametes or between gamete-producing bodies called gametanja, or between hyphae from different strains of fungi. Sometimes both the cytoplasm and the haploid nuclei fuse immediately to produce the diploid zygote, but usually there's a delay between cytoplasmic and nuclear fusion. And this produces what's called a dikaryotic stage, in which cells contain two separate haploid nuclei, one from each parent. So the two cells, for example, are fused, but the two nuclei still remain separate. After a period of this dikaryotic existence, the two nuclei eventually fuse and undergo meiosis to yield sexual spores. So, for example, in the zygomycetes, the zygote develops into a zygospore. in the ascomycetes, an ascospore, and in the basidiomycetes, a basidiospore. To the right here we have a drawing of the generalised life cycle for fungi, and this shows that alternation of haploid and diploid stages. Some fungal species don't actually pass through that dikaryotic stage that's indicated in this drawing. The asexual or haploid stage is used to produce spores that aid in the dissemination of the species, whereas the sexual or diploid stage involves the formation of sexual spores that can survive adverse environmental conditions, for example, such as cold, dryness, and heat. Here we have an excellent illustration of a fungus that's alternating between haploid and diploid stages within its life cycle. In other words, it's alternating between asexual reproduction and sexual reproduction. This is the common bread mold called Rhizopus stolonifer. This fungus grows on the surface of moist, carbohydrate-rich foods such as bread, fruits and vegetables. So on breads, for example, Rhizopus rapidly covers the surface, and hyphae, which are called rhizoids, extend into the bread and absorb nutrients, as we can see labelled here. Other hyphae, referred to as stolons, become erect, and some of these erect stolons produce at their tips asexual sporangia filled with black asexual spores, and this gives the mould its characteristic colour. Each spore, when liberated from the sporangium, as you can see in this image, can germinate to start a new fungus. Rhizopus usually reproduces asexually, but if food becomes scarce or environmental conditions become unfavourable, then it begins sexual reproduction. And sexual reproduction in this particular species requires compatible strains of opposite mating types. And you can see that these two fungal mycelia that are growing on the bread here, the blue one is labelled as the plus strain, and the yellow one is labelled as the minus strain. These are opposite mating types, and they're traditionally referred to as plus and minus strains because they're not morphologically distinguishable as male or female. So when the two mating strains are close together, then hormones are produced that cause their hyphae to form projections called progametangia. And then these mature into gametangia. So if you look firstly at the little boxed area between the blue and the yellow fungi, you can see that contact between their rhizoids that prepares these two fungi for sexual reproduction. Now, if you follow the upwards arrow, you can see the structures that are forming between the blue and yellow hyphae. Firstly, they're labelled as progametangia, and then if you keep following the arrows, you can see the formation of the gametangia. So after fusion of the gametangia, the nuclei of the two gametes will fuse together, forming a zygote. The zygote then develops a thick, rough black coat and becomes a dormant sexual zygospore. Meiosis often occurs at the time of germination, and this is when the zygospore splits open and produces a hypha, and the new cycle begins.